Bismillahirrahmanirrahim. We start the the video 3 of the genetic technology chapter, and this is syllabus 9700, biology A2 syllabus, and this is the last chapter in that according to the syllabus. Now let's talk of the first topic that we're going to discuss is promoters. Now, what are promoters? Promoters are the region of DNA to which RNA polymerase binds as it starts transcription. So let's say this is the gene for a certain protein, say it is the gene for insulin. Now this of course will be present in all humans, and humans are eukaryotes. So what we have to understand is before every gene there would be an area on the DNA. Now please remember this is the DNA. So the DNA strand has an area where you would have the base pairing ATGC, and so this area which is before the gene somewhere upstream is called the promoter. And this area is like I say a passport. Like if you have to travel you need a passport. So for the gene you need a promoter, it says passport. And what happens is that the enzyme RNA polymerase, so there is an enzyme called RNA polymerase that only recognizes this site. So if we want transcription to take place and mRNA to be made, so we have to have a promoter which has to be put into the bacteria, which we are going to genetically engineer. So if I'm going to put the insulin gene for the human human insulin gene into a bacteria, well, I have to put a promoter with it. But the trouble is that if I put this promoter is a eukaryotic promoter. So this is not going to work. So what I need is I need a bacterial promoter which is going to start transcription of its own enzymes, and then maybe it will also make the insulin for me. So please understand is that every gene, every human gene has its own promoter, but if I put the human insulin promoter, then that is not going to work in a bacteria. Why? Because bacteria are prokaryotes and humans are eukaryotes. So we further explain this. I explain this in a little different manner in the further video of this. This is just an overview of how genes are expressed, and the gene is only expressed if both activators are present and the repressor is absent. So we've got these as the activators and this as the repressor. So if you look at the first example, transcription, activators present, repressor absent. Now little or no transcription. Why? Because only one activator is present. And here of course, why? Because activators are present, but then we've got the repressor. The repressor is the one which is going to repress it or stop it. So that gene is not going to be expressed, which happens in our body because every protein does not need to be made. So the protein should be made as need be. It shouldn't be wasted because body's amino acids are wasted to make these proteins. Then this is a very good definition of a eukaryotic promoter, and I would like you to understand it. In genetics, a promoter is a DNA sequence that can recruit transcriptional machinery and lead to the transcription of the downstream DNA sequence, which is the gene for that protein. Location, located near the transcription start sites of genes on the same strand and upstream on the DNA. Size, promoters can be about 100 to 1000 base pairs. If I say this is ATGC, now these are two base pairs. So 100 to 100,000 base pairs long. Function of a promoter: RNA polymerase binding site, initiation of transcription, control by regulatory sequences, control the expression of genes. Expression of genes means that those proteins would be expressed or those will be made, and the body needs them, so those will be made. We shouldn't be making proteins which the body does not need. Now let's try to understand what is a microarray. Microarray is a glass or a plastic slide, and it's only very small. It's only 2 centimeters square. And it has about 10,000 spots or positions. This is one spot, this is another spot, this is another spot, this is another spot. So we have about 10,000 or more of such spots or positions. And on each of these spots, if I just draw this one spot which I've drawn, my drawing is not very good. We have 500 bases in length. So this is, as I've said, A, T, T, A, G, C, and so on and so forth. It will be about 500 bases in length. And we have many of the same A, T, T, A, G, C here on this spot, in one spot. Now what we're going to do is we're going to take a normal cell and we're going to take a cancer cell. So we're going to take a normal cell, let's label that green, and the cancer cell, let's label that red. And what we're going to extract is we're going to extract the mRNA. Similarly, from the cancer cell, we're going to extract the mRNA. And from the cancer cell, when we extract the mRNA, we're going to label it red. And the normal cell, we're going to label it green. So from the mRNA, we're going to make cDNA, and then we're going to label it the color. Similarly, from the cancer cell mRNA, we're going to get the cDNA and label it red. So now we have two colored DNAs. Now we go back to the microarray, and what we do is we mix the normal cell DNA and the cancer cell DNA with this microarray, and we hope that it will attach. You see, if it is present in the cancer cell, then the cancer cell will hybridize. This word we use is hybridize. Hybrid means it's going to, the DNA is going to then pair up with this. If there is a normal cell, then the green will also hybridize. So what do we get? We get different spots of red, or we get different spots of green wherever the you see the normal cell was green and the cancer cell was red. So the normal cell, wherever that is hybridized, we get a green color. Wherever the cancer cell is hybridized, we get a red color. But what if both have hybridized? Okay, then the green and the red, we get a yellow color. So normal or cancer cell, if that hybridizes, you get the different colors red and green. But if both have hybridized, then you get a yellow shade. So this now helps us in understanding which proteins are made in a normal cell and which proteins are made in a cancer cell. If you look at it, mRNA means that this protein is being transcribed, transcription, mRNA. So if in a normal cell we have, say, 5 proteins which are being made, while in a cancer cell we have 7 proteins which are being made. So then we can find out why those proteins are made, and maybe if we can disable the production of those proteins, we can actually find a cure for cancer. So microarray, and then I'm just going to show you a few more pictures and then help you to understand it a little better. This is a picture of that slide or a plastic cover which has those spots on it. It's a very tiny piece. You can hold it in your hand, and this gives you an idea how small it is. And of course, all this is going to be read by the computer. This is not going to be read by humans. It's just going to be read by the computer, and we're going to just place it in the computer and it's going to be read by the computer. Then this is a picture of what we see, the different green and red and the yellow spots. And then of course I've explained this to you here. We've got a cancer cell and a normal cell and the RNA, and then the labeling, and then the hybridization with the probes. The words please understand: hybridize with probes on the microarray. This is another flowchart which explains it very well. I want you to pause it, I want you to go through it, I want you to see, understand that how this is a slightly more explanation. If my explanation previous was not clear, you can understand this a little better. So please pause the video here and have a look. Another flowchart to make you understand this. I hope it becomes more explanatory by these different diagrams that I am giving you. I think this is another very good fluorescent detection DNA chip. The 4 bases ATGC bind, target DNA sequence is analyzed, and then you have attaching the probe, then the probe DNA, and then of course capturing the image with the CCD sensor, identifying the hybridized probes by image processing. So again, a slightly more detail, and of course if you have understood the basic concept, it should be easy in wording the answer if you get a question on that. This is a DNA microarray genome chip picture, and you can see how we've been able to figure out a whole lot of stuff in which we can understand, of course. And yes, the intensity of light also. If the intensity of the orange color or the intensity of the red or the green is more, that also tells us that more of those proteins are being made in that cell. Like for instance, if in a cancer cell we find out that a certain protein is being made in abundance, so the color would be darker, would be brighter, sorry. And if in a normal cell less of those proteins will be made, so it will be a dimmer red color or a dimmer green color. So this is what we need to understand about the intensity of the color. Also tells us that more or less of that protein is being made. This was a question in your Cambridge exam, and I have posted the mark scheme for it, and I want you to understand it and learn the wordings of it. Probes are short lengths of ssDNA. What is ssDNA? Single-stranded DNA. Probes are the ones which we stuck on the glass piece. So probes are short lengths of ssDNA complementary to the alleles being tested for. Many copies of one type of probe placed in each cell. So when we have that cell, we have many copies of that probe being placed in each of the cell. Then number 4, target alleles, DNA made single-stranded. Naturally, we'd have to have single-stranded DNA. So we'd have to denature it and split the strands. So single-stranded DNA made from either mRNA, or of course we split the DNA. Number 5, target alleles or DNA labeled with fluorescent tags, red, green. Then target alleles DNA hybridizes with the probes, the ssDNA, the single-stranded DNA. Then unbound alleles or the DNA is washed off. Well, some of it might not bind because it doesn't have the complementary bases on the probes, and that would be washed off. And then we use laser or UV light to detect the presence of the fluorescence of the hybridized probes. So this is a very good way of learning the wording of your answer. If you maybe have understood what microarray is. I know I would struggle with that, and I would have to learn it. So I can understand that you may be knowing it very well, you've understood it, but you may not put it in the right words so that you can get your marks in the final exam. Now let's come to the next topic, which is gel electrophoresis. In gel electrophoresis, what we do is we take a sort of like a baking tin or a large tray, and in that we put a comb and we let the gel solidify, but the gel is liquid initially, just like to make jelly at home. And then you take the comb out, and what you get here is wells. Wells means khadde. So you get these wells, and then you actually are going to use gel electrophoresis to analyze proteins or DNA. And then you fragment the DNA and you put samples of the DNA in these wells. And you apply current. When you apply current, the DNA fragments move different distances. Some move further away, some move less, some move more. So we get a pattern. And analyzing this pattern then helps us to understand the DNA fingerprinting. So basically I want you to understand first the gel, then applying a current, and then the fragments or the protein molecule moving different distances. Now what are the reasons why these fragments move and how are we going to analyze it is what you need to understand. Now the movement is dependent on a number of factors. Number 1, the first factor is net overall charge. Negatively charged molecules move towards the anode. The positive and positively charged molecule move towards the cathode. So highly charged molecules move faster than those with less overall charge. Then number 1 is the net overall charge. The second is the size. Smaller molecules move through the gel faster. So smaller molecules move through the gel faster. So like a thin person will run faster while as compared to a person who is not so thin. So smaller molecules move through the gel faster than the larger molecules. Then number 3, this was 2, and number 3 is the composition of the gel. Now the common gels are polyacrylamide. This we use for proteins, and agarose for DNA. So the agarose gel is used for DNA, and the size of the pores, you see gel must have some pores in it because it's going to allow the DNA fragments to move. So the pores within the gel determines the speed with which the proteins and the fragments of DNA move. So we're going to use it to analyze proteins, and we're going to use it to analyze fragments of DNA. So gel electrophoresis either for proteins or for DNA. Now here is an example of analyzing and doing gel electrophoresis on proteins. So we've got a normal hemoglobin and we've got a sickle cell hemoglobin. Now there's only one amino acid difference, but what happens is that difference in the amino acid results in a lower negative charge. So they do not move far. So here if you look at lane 1, we have the standard. This is hemoglobin A and this is hemoglobin S. So separation of hemoglobin by gel electrophoresis. This analysis was carried out on a family in which one child had sickle cell anemia. Now in lane 1 we have the hemoglobin standards, and A is for the normal hemoglobin and S is for the sickle cell. Now the lanes 2 and 3 are the hemoglobin samples from the parents. Now both you can see are AS. This is AS. This is also AS. Both the parents 2 and 3 are AS. Now 4, 5 and 6 are hemoglobin samples from their children. Now you can see the person 4 has both A. So he is hemoglobin A, so that means he is normal. He doesn't have any issues. The person 5 has A and S. So he has A and he has S. So he has hemoglobin A and he has hemoglobin S. The person 6 is the one who is affected. Why? Because he only has the hemoglobin S. So he has hemoglobin S, hemoglobin S. Now he is the sufferer or the person who has the sickle cell anemia. So this is one way in which we do gel electrophoresis of proteins. So the result is where proteins have a charge on them, and that charge helps us to either have a more charge or a lower negative charge, and that of course results in the movement towards the anode or the positive. And this of course helps in identifying and in fact finding out if a person is a sufferer or not, or if he is a carrier or not, or if he has the normal hemoglobin. Let's come to the gel electrophoresis of DNA. So first we take the DNA, the required DNA, which is from any source. Either it can be from a hair follicle or it can be if you have a blood sample. And we do this a lot in criminal investigations. So we get the DNA from the site of the crime and we will then take the DNA, and if we need it, if we have very little of it, we can amplify it using the PCR, which is called the polymerase chain reaction. And then we use two different restriction enzymes. If you remember, restriction enzymes cut at palindromic site like GAATTC, or we can have AAATTT. The reason why we use two restriction enzymes is because we want to get shorter DNA fragments. We don't want large DNA fragments, we want many DNA fragments, and we want smaller DNA fragments. So fragments are selected and multiplied. The DNA fragments are put into a gel and separated by an electrical field. Then a radioactive probe is added to bind to the invisible bands of DNA. You must remember, DNA does not have a color. Human cells do not have any color. Anything in it doesn't have any color. So they can blacken on an X-ray film. And now you get this DNA fingerprint in these bands, and you thicker bands and thinner bands, and you get these different distances that they have moved. They have moved different distances, and as I told you before, the smaller the fragment, it moves the furthest. The smaller fragment moves the furthest, the heavier or the bigger fragments will stay towards the wells. So the wells are the ones which we had the gel electrophoresis tray, and then we have the wells here, and then we have the DNA fragments moving. So the smaller fragments would move further away and the heavier fragments would remain here. The larger fragments would remain next to the wells, while the smaller fragments would move away and to the end of the gel. So this is how we would do genetic profiling of DNA. The human DNA contains a large amount of genetic information. It codes for different proteins, and we call that a gene. But a region of DNA that is known to vary between different people is chosen, and these regions often contain variable number of repeated DNA sequences, like for instance AAA, AAATTT. Now this is repeated many times, so this would be called a variable number tandem repeats, VNTRs. Now every person has different variable number of tandem repeats, and only identical twins will share all their VNTR sequences. So DNA can be extracted from almost anything that has come from a person's body: the root of a hair, the hair follicle, a tiny spot of blood or semen at a crime scene, or saliva where someone has drunk from a cup. Usually the quantity of DNA is increased by the PCR or the polymerase chain reaction, which makes many copies of the DNA. Then the DNA is chopped into pieces using restriction enzyme known to cleave it close to the VNTR regions. Now the DNA is ready for electrophoresis. When the current is turned off, the gel contains DNA fragments that have ended up in different places. These fragments are not visible straight away, and then of course we talked about how we would try to make them so that we could see them on a piece of. So you add radioactive probes, like we just discussed in the previous film, is that you added radioactive probes to bind to the invisible bands of DNA so that they can blacken on an X-ray film. So we have now got a gel electrophoresis of DNA. Here is the application of this gel electrophoresis. Now you can see here we have a controlled DNA, we have the DNA from a mother, and then we have the DNA from the child, and then we have a possible father. Now whenever this is sort of paternity testing, the child will share genetic material with both the mother and the possible father, showing that it is highly probable this is the actual father. Why? Because all the bands in the child will be present in the father or in the mother. So the child must get his band either from his mother or from his father. So when you look at the child's band, they are either from the mother or from the father. So this of course helps us in all these sort of cases in which we have a dispute over paternity. So this is another one. Then another example is this one, in which we have a blood stain at the scene of the crime, and we have people 1, 2, 3, and then 4, 5, 6, 7. These are the 7 suspects who might have committed the murder or the crime. And so DNA fragment of a blood stain found at the scene of a crime compared with fingerprints of 7 suspects. So this is the way that we use this gel electrophoresis, and this is called DNA profiling in crime scene analysis. So the applications of gel electrophoresis are vast, and of course they are very, very important in the field of biology.